Okay, let's move ahead a little bit. We're going to talk about the juxtaglomerular apparatus. The juxtaglomerular apparatus is a microscopic structure in the kidney which regulates the function of each nephron. And there are three cellular components of the apparatus, which are the Macula Densa, extraglomerular mesangial cells, and the Juxtaglomerular cells, also known as the granular cells. Let's talk about those in a little bit more detail. So as I was saying, the juxtaglomerular apparatus is a microscopic structure in the kidney that regulates the function of each nephron. And it is named for its proximity to the glomerulus. It is found between the vascular pole of the renal corpuscle and the returning distal convoluted tubule of the same nephron. And this location is critical to the functioning in regulating renal blood flow and glomerular filtration rate. And as I said before, the three components, the macula densa of the distal convoluted tubule and the smooth muscle cells of the afferent arteriole are known as juxtaglomerular cells, and then there are also extraglomerular mesangial cells. So let's discuss those. First, we can discuss the juxtaglomerular cells. They're also known as granular cells, and they are the specialized smooth muscle cells of glomerular arterioles, and they produce and secrete renin. Next is the macula densa cells. These are columnar epithelium thickening of the distal tubule. The macula densa senses any increase in the sodium chloride concentration in the distal tubule of the kidney and secretes a locally active paracrine vasopressor, which acts on the adjacent afferent arteriole to decrease glomerular filtration rate as part of the tubulo-glomerular feedback loop. And finally, we discussed the extraglomerular mesangial cells. They're also known as Lassis cells or Gore-Magdic cells or Polkisson cells, and they are light staining cells in the kidney found outside the glomerulus near the vascular pole and macula densa. These Lassis cells form the juxtaglomerular apparatus in combination with two other cells, as mentioned, the macula densa of the distal convoluted tubule and the juxtaglomerular cells of the afferent arteriole. This apparatus controls blood pressure through the renin-angiotensin-aldosterone system, and the specific function of the Lassis cells is not well understood, although it has been associated with the secretion of erythropoietin. Let's talk a little bit about the macula densa. The cells of the macula densa are sensitive to the concentration of sodium chloride, as said before, in the late thick ascending limb, and a decrease in sodium chloride concentration initiates a signal from the macula densa that has two effects. One, it decreases resistance to blood flow in the afferent arterioles via vasodilation, which increases glomerular hydrostatic pressure and helps return glomerular filtration rate towards normal. And two, it increases renin release from the juxtaglomerular cells of the afferent and efferent arterioles, which are the major storage sites for renin. And as a result, an increase in sodium chloride concentration would result in vasoconstriction of afferent arterioles and reduced paracrine stimulation of juxtaglomerular cells. This demonstrates the macula densa feedback where compensatory mechanisms act in order to return the GFR to normal. Thank you, Wikipedia. And again, just to make sure we understand the juxtaglomerular cells, they're also known as JG cells or granular cells, and they are cells in the kidney that synthesize, store, and secrete the enzyme renin. And they are specialized smooth muscle cells mainly in the walls of the afferent arterioles and some in the efferent arterioles that deliver blood to the glomerulus. In synthesizing renin, they play a critical role in the renin-angiotensin system and thus in renal autoregulation of the kidney. And the juxtaglomerular cells secrete renin in response to a drop in pressure detected by stretch receptors in the vascular walls or when stimulated by the macula densa cells. The macula densa cells are located in the distal convoluted tubule and stimulate juxtaglomerular cells to release renin when they detect a drop in sodium concentration in tubular fluid. Together, juxtaglomerular cells and the macula densa cells comprise the juxtaglomerular complex. Okay, it's very interesting.